Well, good morning, everyone. I'd like to thank you all for listening. Today I'm going to talk about this really exciting new technology that has just kind of jumped onto the scene in the last 5 years, and it's known as CRISPR. And so this is really not only being used in pretty much every lab in the world, but it's kind of caught the greater imagination of the wider community as well. So we've seen it on the cover of Time magazine. It's been called Breakthrough of the Year. This laser's not great. And we're seeing it being used in more and more places, to the point where it's caught the imagination of the dystopic kind of fascination that Hollywood has with science and science fiction. So there was this recent movie where there were large gorillas that were accidentally made in a CRISPR experiment gone wrong. And so it's been interesting to be part of all of this. And the big question is, what is CRISPR? And so following on from Hollywood, I'll let Hollywood tell you what they think CRISPR is. And if we can get this video to work. And so
we have to figure this out. What's wrong with the science?
Okay. The Spartan virus removes the adenosine deaminase gene from your DNA. Remove the ADA gene and your immune system will simply vanish.
Yeah, but I'm not getting sick.
It's only a matter of time. Without alien DNA, you don't stand a chance against the Spartan virus.
Okay, so how does it work? How does the virus remove the ADA gene?
A process called CRISPR-Cas.
CRISPR-Cas9. RNA and a protein cutting genes at exact locations.
Exactly.
And they got that exactly right. So what CRISPR is a technology to be able to precisely edit and cut the genome at any location that you desire. And so this has been used in a range of organisms and also in us. And so I don't want to play that again. Where did CRISPR come from? It wasn't a tool that was invented by humans, but it actually comes from an immune system evolved in bacteria and archaea. And so I'm going to take you through and explain how people came to understand this as a potent tool for modifying genomes. And so it was many years ago, people, the year I was born actually, people discovered that there was this interesting repeat-like sequence in the bacteria of E. coli. These are common bacteria that we use in the lab all the time for molecular biology. And if you zoom in on the genome of these little bugs, they found these really interesting repeat sequences that we now know as CRISPR sequences, or clustered regularly interspaced short palindromic repeats. It's got a mouthful, so that's why we've got this nice acronym. And what was interesting about this wasn't only the repeat-like structure, which often exists in a number of genomes for different reasons, but it was the identity of the sequences between these repeats. And so when people looked at these sequences, they didn't look like bacterial sequences, but they looked like viral sequences. And so this was quite odd. And over many years, people kind of hypothesized that maybe bacteria are collecting these viral sequences and storing them as a kind of molecular memory, as an adaptive immune system to protect themselves from viruses. So pretty much every organism on the planet has to protect themselves from viruses, and bacteria are no different. And they have the ability to protect themselves from what we call bacteriophage, or eaters of bacteria. Now, interestingly, these are the most abundant biological entities on the planet. There's more phage on the planet than bacteria tenfold. And so the number's quite staggering. If you take a mil of seawater, there's 50 million bacterial viruses in that seawater. And so they're naturally all around us. They're in our gut. They actually contribute to our gut health as well, which is a burgeoning area of research. But just like any organism, organisms need to protect themselves against these kind of infections because they're quite catastrophic. And so the early hypothesis was that maybe CRISPR's an immune system. And so this was kind of on the fringe. There was one scientist in Spain that had been working on this for many years and kind of unnoticed. And it really wasn't until a yogurt company came along, Danesco, now part of DuPont, and became interested in this because they were trying to grow large amounts of Streptococcus thermophilus for yogurt production, but their bacteria kept dying. And so this is a common problem in industrial biotech. and they wanted to work out new ways that they could immunize their bacteria. And so through their research arm, they actually found that they could actively immunize their strep strains using this CRISPR system. And so this was really the first experimental evidence that this system was an immune system. And so it's much like a vaccination card that we used to get back before computers, where you would basically tick off the list of all the things that you're immunized against. And so how does this system kind of work at the molecular level? And really what CRISPR is, in many cases, a set of molecular scissors that uses, as the X-Files says, a protein molecule and an RNA molecule that then can be used as a homing device to really go in and target the viral DNA without cutting your own DNA. So adaptive immunity that is able to recognize alien DNA very much. And so a number of these CRISPR systems exist, and the main ones that we use for genome editing obviously cut DNA, but we also have ones that cut RNA. And that's what I'll talk to you about at the end of my talk, and that's what my lab's particularly interested in, is targeting this molecular cousin of DNA. And so they basically were able to cut the DNA, and in bacteria, once you cut viral DNA, they're no longer able to replicate and the virus is destroyed. And so in a kind of more graphic kind of version of this, you have these bacteriophage that really want to amplify their own existence. That's the whole reason why viruses exist. They're just selfish genetic elements. And so they inject DNA into the bacteria, but the bacteria are ready to attack this DNA through the CRISPR system. So they have these RNA molecules that then form a complex with with a protein in this case, known as Cas9, and then this guy will float around and we'll be able to use the base pairing between the RNA and the DNA to act as a molecular recognition, kind of like a zipper. And if this zipper matches, you end up with a destruction of the DNA. And so now we know that how it works. It's this very precise molecular scissors that's able to cut DNA. What can we do with it? So a lot of scientists are always looking for new ways to manipulate DNA, and phage biology and bacterial biology for many years has given us lots of things that we use in the lab every single day, whether it's PCR enzymes for amplifying DNA, or now CRISPR for being able to cut DNA in very specific ways. So bacteria use it to cut viral DNA in very specific ways. Can we use it to cut human DNA or any other organism to edit DNA to treat genetic disease or give crops favorable traits? And so in bacterial cells, you lead to degradation. But in human cells, we have, and other higher eukaryotes, we have sophisticated repair mechanisms that once we cut a bit of DNA, it gets repaired by other cellular proteins. And we know a great deal about how the molecular biology of how this repair process works, and so we can harness that to actually repair pieces of DNA. And this has been done in labs all across the world. And so you can really think about it as a kind of find and replace kind of function in a Word document, where you might have a spelling mistake, and you can find that mistake and then using the endogenous machinery of the human DNA repair, replace it. And so in this case, we're looking at large bits of DNA sequence. We're really able to home in on this aberrant T here and be able to change it now. And so this all came about from understanding how these nucleases, as I said, work in bacterial immunity. And so this was published in 2012 in the lab that I used to work in, and rather rapidly it was shown to be able to be adapted for use in human cell culture. And just in the few short years, pretty much every organism that people can get their hands on, they're now being able to CRISPR all kinds of things. And many of these are being used either in research or in agriculture or in our foods. And so it's really kind of rapidly been adopted by the wider community. And this is mainly because it's low cost. There were genome editing technologies that have existed for the last 20-odd years, but they were quite hard to use, and they were mostly restricted to labs that had extremely extensive resources to carry them out. But CRISPR's kind of democratized gene editing for pretty much anyone that can get their hands on a CRISPR enzyme. And so it's been used in anything from basic research to disease modeling, and the agriculture companies are really have pretty full-blown CRISPR programs now to create new varieties of crops that aren't genetically modified the old-fashioned way, and so much more precise and safer in that respect. And so what we're really interested in as scientists is, can we cure genetic diseases at the level of DNA? So as many of you know, there are multiple diseases that are caused by just small changes in the DNA, and things like sickle cell disease or cystic fibrosis. And now these have become much more tractable therapeutic avenues using CRISPR to potentially cure these things. So this kind of whole field of research has really blown up in the last couple of years, and there are hundreds of papers out there now, publications showing proof of concept of being able to cure these, at least in the lab or in model organisms. And here are just a small sampling, so anything from cystic fibrosis to hemophilia, or even editing viruses to cure some of the latent infection problems that we see with some viruses. And so genome editing has already been successful in the clinic, and this has been using older technology that was developed before CRISPR, but we're really going to see an explosion with this new technology because it's a lot easier to use. And so there's one study done by Sangamo Biosciences out of California where they were able to take human T cells and remove them from the body and edit a receptor that's required for HIV infection, and then put them back in HIV patients, and effectively it's a cure for HIV. So an expensive method, but it's able to basically rid most of the cells of HIV, which is pretty amazing. And there have been several other cases using, similar to what our previous speaker was talking about in terms of immunomodulation using gene editing. And so several companies have now started that are really tackling this head on, and they're tackling all kinds of genetic disorders in hope to cure them once and for all. And so what I really want to emphasize here is that this kind of obscure research into a bacterial immune system, done kind of in the fringe of science for many years, really led to what we see as a revolution in the ability to precisely manipulate genomes in all organisms and for health and for human food security. And we're really still tackling the incredibly diverse variety of different immune systems that exist in bacteria and archaea, and it continually is amazing us about what we can find. So what I was talking about before was this single enzyme, Cas9, but CRISPR systems contain hundreds of different types of enzymes that we really don't understand how a lot of them work. And so this is really what my lab's excited about, is taking some of these newer enzymes, whether they're different versions of Cas9 or these completely different molecules that have similar properties, and using them to manipulate gene expression in new and interesting ways. And so to sum up what we do, it's really, we're at the interface between developing new technology as engineers and as RNA biologists, really interested in, as Dr. Tubman said, being able to manipulate gene expression in disease where RNA regulation goes awry. And so you probably remember this from maybe your first biology class in high school, but RNA is really an intermediate between DNA and protein. RNA also does a lot of other stuff that's not readily linked to protein production. But what's fascinating even about this in the simple case is that beyond the requirements for RNA to code for protein, there's lots of other regulatory sequences within the RNA that only controls where the RNA gets made. So in neurons, for example, the RNA gets shuttled out to the synapses, so the protein can be made in a localized location. How long the RNA sticks around for, not all RNAs last the same amount in a tissue. And if we can target RNA in a precise manner, we should be able to manipulate some of these processes that often go wrong in disease, and it really offers a new opportunity for research and therapies. And so what I mean by that is if we have a molecule such as Cas9 or one of these newer ones, Cas13, we can park it on a particular RNA and then start to carry out a range of different molecular functions, whether it's looking at where an RNA is in the cell, understanding its localization, chewing up an RNA, so it would downregulate its expression, or many other functions. And so I'll give you one thing that we're particularly interested in at the moment is being able to manipulate a process known as RNA splicing. So when a gene gets expressed in a cell, from a DNA locus within the cell, it gets made into what's known as unprocessed pre-mRNA. And for this mRNA to become an active mRNA that can be translated into protein, all these little bits of RNA that need to be cut out of the immature RNA, and then the RNA needs to get joined back together to then be read by the ribosome to make protein. And what happens, particularly in more complex organisms, is this splicing mechanism can happen multiple different ways, and this is known as alternative splicing. So in some cases, you might have all of the exons, all these different bits joined together in the right order. Sometimes things might be skipped out. Other times, additional pieces of RNA might be put in. And what this results in is, from one gene, we often have multiple different protein isoforms that are made. And this can affect things from cancer to signaling cascades, and even modify the ability for drugs to work. And we really don't understand on a genome-wide scale what these protein isoforms are doing. So if we had tools to manipulate this process, it would be really powerful. And there's a long list of different diseases, and I'm not going to go through them, but a lot of dystrophies and spinal muscular atrophy is a very classic case where splicing goes wrong. And furthermore, in cancer, there's a growing understanding that most mutations in cancers are actually disrupting this splicing process, leading to proteins that don't function properly and can lead to transformation and metastasis. And so our idea is that in some cases you might have too much of this toxic protein isoform B being made, and we really want to shift the balance back to the protein isoform A that contains this additional sequence. And one way to do this would be to take our CRISPR enzymes and synthetically tether them to splicing factors to make this a change, to really force this change in the expression of this protein. And so I'm just going to show you a little bit of data that we've recently generated. It's unpublished, and in a collaboration with some colleagues at UC Berkeley, where we're taking these CRISPR proteins and synthetically making these artificial transcription factors. So a lot of these proteins, you don't really need to know the details, but these are involved in regulating splicing naturally in the cell. So we're co-opting these and synthetically attaching them to CRISPR to create synthetic splicing factors. And so we can create a range of different factors, and what the idea is, can we start to manipulate this natural process, particularly when it goes awry in cancer? And so we have some very preliminary data to show that when we park a Cas9 molecule here with a special splicing repressor domain fused to it, and we have to park it in a very particular location within this intron, we get a nice decrease in the amount of exon that is included. So this is just a nice proof of concept that we can target a particular RNA in the cell, and these are in human cell culture, and be able to reduce the amount of exons that are included. And so now I'm going to switch to another enzyme that we've become particularly interested in as a biotechnology tool, but here we're using it for a very different purpose. So a couple of years ago, I came up with this idea to be able to use this enzyme that has this peculiar enzymatic activity to detect very small amounts of RNA. And the way this works very briefly is that you have an RNA molecule that you're interested in. You can design this CRISPR molecule to bind precisely to this RNA molecule, and once it's bound, it becomes this very active enzyme that's able to use this chemical as a substrate to create fluorescent. So every time we see very small amounts of RNA, this enzyme will tell us that it's there through some fluorescence. And what we were able to show as a proof of concept is we could just take RNA from human cells and detect a very specific RNA that's only present in human cells but not present in total RNA from E. coli. And so this is a one-pot kind of reaction where we can detect whether RNA is present or not. And now this technology has been used by other labs to detect very, very small amounts of RNA in a test tube using an extended approach. And without going into the details, what's really exciting about this is they've taken this fluorescent detection approach that we developed and added a few extra steps, but able to basically print it out on a pregnancy test strip. So they've got it to the point now where they can take this, our detection approach, and go out in the field in Africa and within as little as an hour take some saliva from patients or healthy samples and be able to detect whether Zika is present in their saliva. So it's kind of this a fascinating thing that this obscure enzyme from bacteria can then be rapidly adopted for some kind of in-the-field technology to detect Zika in the field. And so we're really excited about where this is going in terms of future technologies for the detection and manipulation of RNA. And so people are beginning to think about using this, and there are a number of companies that are now developing this technology for not only viral detection in the field, but also detection of cancer-related mutations in DNA. And these testing approaches are rather cheap. So one of these strips costs 50 cents to make, so it's not an obscenely expensive process. And so to finish, I'd just like to, science is done as part of teams, so I'd like to really acknowledge all the people that I work with. This is my current lab, so I've only been here for a year, but we've really grown quite rapidly, and I'm really excited about all the new science we get to do. And I'd also like to thank a lot of the people that I used to previously work with when I was in California, and I'm really fortunate for all the help that they've given me. So if you have any questions, I'm happy to take them.
Would you mind commenting on the ethical issues regarding CRISPR for altering human genome DNA, please?
So that's a great question, and I actually had some slides on that, but I decided to take it out because it's a minefield. So there's two ways that CRISPR can be introduced. So you have somatic cell engineering. So you could take a patient, you could engineer their cells, and that's not going to be passed on to the next generation. And I don't think anyone has many ethical issues with that as long as it's used for appropriate health concerns, such as genetic disease or things like that. Where it gets a little more dicey, and I don't think there's a big consensus on this, and it depends on the nation that's doing it. So China's doing a lot of this already, is germline engineering, where you can edit an embryo, which is effectively pretty easy to do because there's only a couple of cells, you can get a pretty high hit rate, and that is passed on to future generations. So I think ethically there's a school of thought that says if we don't cure genetic disease and we hold this technology back from people, that's unethical. But there's also a school of thought where is the line, particularly when it comes to making changes that can make people faster runners or have more muscle, and then it becomes a question of between the haves and haves not. And this is going to be an expensive technology to implement in those cases, so it could further produce inequality. So in the US, there's been a lot of talk around this, and there's been a lot of regulatory guidelines set up, and we're not allowed to do germline engineering yet in the US on viable embryos. So I think people are really concerned about where this is going, but other countries have a lot less regulation. So
That's the issue with technology. Generally,
yeah.
very higher aim.
Yeah.
And this is unfortunately what usually happens. CRISPR gives us a chance to change that three-word phrase.
Yeah. I think this time there's been a lot more upfront regulatory discussion before the technology's got ahead of itself. So I think we're in a better space this time around.
Other questions?
Thanks very much.
Thank you. I'm the Senior Associate Dean for Clinical Research. He's also a co-director of the CTSI and one of the principal investigators of the CLIC grant I just mentioned. And Rich Libby, who is the new Senior Associate Dean for Graduate Education. We also had a new leader this year in terms of the medical center, Karen Davis, who became our Chief Nursing Executive, coming from the Johns Hopkins system, and we have been thrilled that she joined us. Just to give you some idea of the vibrancy, so since Pat Brophy got here in January as chair of pediatrics. These are the new recruitments to the faculty of pediatrics. And I wanted to get, I think we have a similar slide for psychiatry, but we couldn't get it in time, and I think this makes the point that we are really recruiting increasingly extraordinary chairs, and this is becoming a increasingly desirable place to work. So it gives you a little bit of a sense of our research enterprise. We are probably very different. For the 50-year class, I don't think you could have envisioned it. I got to tell you, I'm here 16 years. I don't think I can envision this. But as is true around the country, we're now a pretty large healthcare system. We call that broader system UR Medicine. It involves 6 hospitals, not only Strong and Highland, but hospitals in Jones, Noyes, and St. James in the southern tier, F.F. Thompson in Canandaigua. But we also now have 225 outpatient clinics. We have 8 urgent care centers, and as you can see, we have over 3 million outpatient encounters a year. This is what this footprint looks like on the map. We have what we call our Accountable Care Network, which we call AHP, Accountable Health Partners. That now comprises 2,000 physicians, roughly 1,350 from our employed medical faculty group here at the University of Rochester. The rest of our community physicians. And you can see it is split between adult primary care and pediatrics, OBGYN, and probably close to 75% are specialties. Our network extends broadly throughout upstate New York. These are where our affiliates is. This is where we do virtually all tertiary and quaternary care and some secondary care. But we also now have collaborations with other health systems like St. Joseph's in Syracuse, which is part of Trinity and has 200,000 lives. The Bassett Health System in Cooperstown, another 200,000 lives. So this gives us a network of over 1 million patients, half of whom we are primarily responsible for, the other half where we are getting referrals that support our specialists and really allow the University of Rochester to have a mission that is bipartite, meaning we don't want anybody to ever have to leave upstate New York to really get the highest end, cutting edge, state-of-the-art therapy, whether it's CAR T-cell therapy, RNA-based therapies in any disease. But we also want to provide a high quality of standard locally. So we're trying to build up programs in all of these regions so that most patients, 98% of patients, can be cared for as close to home as possible, leaving our hospital, which is running for the last 5 years at over 105% occupancy for medical and surgical beds, leaving them really to take care of the people who can only be taken care of in a university setting. I mentioned our urgent care network. That has expanded over a 3-year period. 5 years ago, we had 1 urgent care center that had 10,000 visits. This year, we're up to 70,000, which is very much important for patient satisfaction, allowing them to get care at night and off hours, and to some degree also trying to decompress our emergency room a little bit. This is an example of what we look like as a system. So these are all the branches of the Wilmot Cancer Institute, which goes as far as Elmira in New York. And in fact, we opened up a new building, a branch of the Wilmot in Dansville. And again, the goal of this is not only have we now become one of the 25 largest by volume cancer institutes in the country, but it allows us to provide the same quality of cancer care at home. So patients in the southern tier, which is a large, very poor area, do not have to schlep into Rochester to necessarily get infusions or radiation therapy they can get it close to home. But this is somewhat of a model that you're going to see reproduced in other specialties as well, but it's probably most well-formed at the Wilmot. Because of this growth, and we now are 50% larger than any other cancer center outside of New York City, one of our other key goals in terms of research and clinical is to gain NCI designation. We have a plan to submit our application for 2021. For this, we have several high-quality programs because you need 4 priority scientific programs. And we already have one of the great survivorship programs in the country. This year, we were able to recruit Paula Vertino from Emory University. She is an epigeneticist who became our first Wilmot Distinguished Professor and the Director of Translational Research for Cancer. So this is moving forward, but it's a major goal for us. One of the things I should mention is that between 12:30 and 12:45, you can see the mobile stroke unit. So this month, actually, we became one of only 12 in the nation to set up a mobile stroke unit. What this is a unit that is equipped with a CT scanner, which can wirelessly transmit the CTs to the U of R. We are working with the ambulance, the emergency system in Rochester, so that when EMTs are called and it is suspected that it is a stroke, they can then send out for the stroke mobile, can come, and unless they're very close to the U of R, they can actually get their CT scan in the mobile, and we can get the results, and if they're far enough away, we can actually start therapy while they're in the stroke mobile. This has been done in a few cities and has drastically improved the outcome of stroke, because obviously in stroke, the faster you can intervene, the less neurologic damage. And so this is something that initially will be in a 45, 50-mile radius, but we hope that we will be able to expand this more broadly. We also, as part of a network, actually this year developed a collaboration with the YMCA. And this just gives you a map to gives you some idea. This is broad. It's throughout the region. And what this will, again, allow us to do is to develop programs in 17 YMCAs across the region. And you can see what some of these are. They'll be targeted in specific areas, again, so we can bring more care locally. So people can get care, get education, sometimes get screening locally in the communities. I'm not going to say much about this, but except that, as you can imagine, given the growth of the system, we're seeing huge growth in surgical discharges from Strong, from Highland Hospital, again, where they're most often above 100% occupancy. We are increasing our outpatient encounters almost by 200,000 a year. Our emergency department continues to grow, and our outpatient surgery has grown to the point we've actually now maxed out. So what this means, to quote Jaws, is we got to build a bigger boat, or we have to build bigger boats in order to provide be the regional network and provide the care that we need. Among the things that we are really formalizing this year are plans for 2 buildings. One is a new building. This is our current emergency room. This is the entrance to the hospital. So here is where we envision the building. So it'd be to the right of the emergency room if you're looking at it. This will increase emergency room space by 3-fold. It was originally built for 50,000 people. We're now seeing 125. Will allow it to be all private rooms and with specialty areas, and then will allow additional floors, which will both allow us to expand our beds, but more importantly, convert all the beds at Strong and in this tower to private rooms. I hope all of you will get a chance to look at some of the buildings that have already come up. So we have the Golisano Children's Hospital and the Cancer Center, both of which actually are all private rooms, including what I think is one of the most extraordinary things is our neonatal ICU, which also has private rooms so that parents can stay with their preemies 24/7. We're also building an orthopedics building, which will not only expand the number of operating rooms, number of exam rooms by about 50%, but will allow us to move all of the orthopedic operating rooms to this building, and will create a sports medicine campus with a field house because we now have a destination sports medicine program for upstate New York. This will allow us to use our current outpatient ORs for everything but orthopedics. So these are two buildings that will help us provide services to the region. Finally, last but not least, I just give you a snapshot of what we look like. We are now this medical center is about a $3.7 billion operation. It has an actual operating margin of roughly $66 million, a little bit more than we anticipated. But to understand that margin, this is after both support of the university, but also after $60 million of investment in research and education. And what we have actually done is to move into a fully integrated budget so that we make sure that we can adequately provide for our three great missions. And lastly, and very importantly, and for which we owe the people in this audience a debt of thanks, we had another great year in terms of advancement. I think this is particularly important because we are not in the middle of a capital campaign yet, but so you are worried that when you don't have a campaign, it will go down. It actually went up. We actually had, as you can see, about $68 million in cash, $76 million in commitments. This is the second largest we've done since even in the campaign. We've also now have a more vigorous patient family giving program. And we owe it to many of you in this room who have helped support this advancement. Applications which, during this campaign created 65 endowed professorships, helped support the two buildings, or all the buildings that you see on Crittenden Avenue, and hopefully will allow us to keep moving forward in our research enterprises. I want to congratulate today's awards winners, most of whom received their awards at the Whipple dinner last night. It was an exciting dinner. I know many of you were there. And to mention in particular that at 1:30 we will have Phil Greenland, who received the Distinguished Alumnus Award, will be giving his lecture. Finally, I think we have show that there were two. Again, I wanted to mention that you should be able to look at the there it is, the stroke mobile, and please do take a look.
I'm Ruth Anderson Lawrence, the class of 1949. I'm sitting in the very spot I sat in 1945 when I came to medical school, and my class met with George Whipple for our very first gathering. I'm excited to take you on a tour of the historic and important locations in the School of Medicine and Dentistry and the hospital. We call it the Heritage Trail. I hope you enjoy it. Our next stop will be Whipple Auditorium on the second floor. Well, this room was one of the original rooms in the medical center when it was built in 1925. And every class, their first day, assembled in this room and met the dean, Dr. George Whipple. This room was cement block walls and very uncomfortable seats. They were wooden. Now we head to the first floor to see the minor library. As you enter the library, walk straight back to the reading room, which is the original main entrance and lobby of Strong Memorial Hospital, and where the original fireplace still remains. We used to come in the front door of the hospital, one flight down, and walk up this very elegant set of stairs, and you'd be in the lobby of the hospital. These wonderful walls are just the same, and probably the thing that every student remembers is the fireplace over there. And it was always going. Our next point of interest is the original office of George Whipple, the school's first dean and a Nobel Prize recipient. He occupied the office from 1953 to 1976. It's now called the Whipple Museum. After his death in 1976, the decision was made to memorialize Dr. Whipple by leaving his office just as it was when he passed away. To this day, the books, the papers, and the personal items in the office lie in the exact spot in which he left them. This room looks just like it did when I was interviewed in 1945. Directly across from where the old bookstore used to be, on the ground floor, is the original entrance to the School of Medicine and Dentistry. If you go outside, you can see the 4 columns. Our final stop is the original student mailboxes. As you can see, the mailboxes are the same, only their location has been changed.
So I hope you will join Ruth Lawrence for the Heritage Tour. And again, I want to thank you for coming here this year for your reunion. There'll be lunch, there'll be lectures. I hope you also will take advantage of the lectures and events at the River Campus as part of the Meliora Weekend. There are a lot of exciting adventures. I know I've got my tickets here to Pink Martinis. I'm not sure what pink martinis are, but people tell me that I'm going to love them. So thank you all for coming, and thank you for what you do.